Hello everyone, thanks for clicking on my video. Enzymes play an important role in human life. This video will learn more about enzymes by focusing on how to measure lactase activity and about lactose intolerance. First, I will give some background information ahead. So first, what is enzyme? Enzymes are biological catalysts with remarkable power, increasing reaction rates by at least a millionfold. They are usually proteins, although some enzymes are other types of biological molecules. Enzymes function best under limited conditions of temperature, pH, and salt concentrations. Enzymes are highly specific as to substrates and reactions catalyzed. Some enzymes require one or more non-protein components called core enzymes if they are organic, and core factors if inorganic.
Then, what is activation energy? Activation energy refers to the energy barrier that must be overcome in a chemical reaction in order for the reaction to start and continue. Here we use Mario as an example. If you think of Mario's progress through the level as a reaction, and Mario has to jump over a growing obstacle for the reaction to proceed, then the growing obstacle here is like the activation energy. So how do enzymes work? The enzymes lowers the growing obstacles, making it easier for Mario to jump them and complete them faster. In other words, enzymes speed up the reaction by reducing the activation energy of the reaction.
In this experiment, lactase was selected as one of the experimental materials, which is a very common enzyme with highly conserved activity in the evolutionary process. Lactase catalyzes the hydrolysis of beta-glycosidic bonds in the disaccharide lactose, releasing galactose and glucose. It's important for humans to digest milk. So how lactase activity is measured? First, we will extract lactase from dietary supplement tablets and measure its activity under different conditions using the spectrophotometry. The spectrophotometry is a quantitative analysis technique widely used in chemical analysis and biochemical experiments. It is based on the principle that matter absorbs or emits electromagnetic radiation of specific wavelengths. To do this, the substrate or reaction product must absorb light, but none of the molecules can. To solve this problem, we are going to attach another molecule to the glycoside that mimics the shape of lactose. We called ONPG here. Since ONPG can produce color reaction, this experiment will explore the amount of nitrophenol produced by enzyme reaction within a fixed time by measuring the change of absorbance. Then we will introduce the reagent supplies and equipment used in this experiment. So first, we will use a commercial lactase tablets, mortar, and pestle. Also, the micropipettes tubes dry best at 37 degrees centigrade, and also the microcentrifuge and tubes, microplate reader, and a 96 well plate will be used. Then we will also use 1 tube of PBS, 1 tube of ONPG, and 1 tube of sodium carbonate.
Then we come to the procedure part. First, crush 1 lactase tablet containing 200 mg of enzyme to an even powder using a mortar and pestle. The resulting powder is transferred to an winning paper folded diagonally and transfer the powder into a 15 ml tube labeled as suspension. Then 1 ml of suspension was transferred into two 1.5 ml centrifuge tubes labeled with lactase suspension.
Then centrifuge them at a rotational speed of 10,000 rpm for 1 minute to precipitate solid particles. It was noted that the centrifuge tubes should be placed opposite each other. Then 500 microliter of the supernatant was transferred from each tube into a 1.5 ml tube labeled lactase extract.
In the next part, first prepare 14 1.5 ml tubes and label them according to the table on the right. Add PBS buffer, OMPG, and lactase extract in each tube according to the table. Tap them and mix them immediately. Place it in a dry bath set at 37 degrees centigrade. Time according to the time in the table and culture at the required temperature. Immediately remove and add 500 microliter stop solution to stop the reaction when the time is up. After all operations according to the table, pipette 200 microliter from each tube to the wells of a 96 well plate and measure the absorbance at 450 nanometer. Meanwhile, a brief description of the experimental results is given here. After obtaining figure on the right, the enzyme activity value can be obtained by fitting the slope of the straight line, and it is concluded that the longer heating in the range of experimental time, the higher the reaction degree will be under the set temperature in this experiment. At the same time, it is also hoped to expand the knowledge of enzyme activity experiments and related it to the real world. As mentioned earlier, lactase breaks down lactose, which is one of the main nutrients in milk. When the human body lacks enough lactase, it cannot effectively break down lactose from the components, which will result in the lactose intolerance. And this is more common among Asians. At the same time, the causes of lactose intolerance are also divided into three conditions. The first one is primary hypolactasia, mainly related to genetics. And the next one is secondary hypolactasia, mainly caused by damage to the small intestine. And the last one is primary congenital lactasia, mainly caused by a genetic defect in which patient may not be able to digest breast milk. So what are some obvious symptoms of lactose intolerance? When ingested, lactose is not fully digested and stay in the intestine. It can cause bloating, abdominal pain, diarrhea, and even vomiting. But there are also some ways to deal with lactose intolerance. The first one is the zero lactose milk, which reduces the lactose content of milk and ensures adequate intake of other nutrients and calcium from milk. Secondly, lactase supplements can be used with food to avoid lactase deficiency. Finally, seeking medical advice can also be an important help in addressing lactose intolerance. Under this method, even if there is a lactose intolerance, you can try to drink special milk or eat other food to replace milk to supplement the required nutrients. At last, thank for your listening and thank Professor Lee for giving us a foundation of biological knowledge. And also thanks for my best single pair share group 10 for the best discussion in class and help me solve my confusions.